Thank you for the organization of this wonderful webinar. There were lots of interesting talks yesterday and seems to be quite the same for today. And thank you for providing me with the opportunity to present a really small part of one of our projects, which is dedicated to the structures, the studies of the structure and the inhibition of enzymes that are called Phosphopantetheinyl transferase, which I will refer to as PPTase. It's much more easier to say. And I will more specifically focus today on one specific enzyme, which is the PPTAB enzyme from Mycobacterium tuberculosis. So usually when we speak about mycobacteria, we often refer to Mycobacterium tuberculosis. It's one of the most known mycobacteria. It's one of the deadliest pathogen worldwide, which claims about 1.5 million deaths every year. There are 10 million new cases of tuberculosis every year, and it's very often resistant to at least one drug, which is used in anti-tuberculous therapy. Okay, I will not talk about tuberculosis today. I will talk about a cousin of this mycobacteria, which is called Mycobacterium abscessus. It's a little bit less known, but its prevalence is increasing a lot, and it causes skin, lungs, and soft tissues infection, and it's often affects people that are already dealing with cystic fibrosis. And what is really bad news is that in about 50% of the case, the antibiotic will not be able to treat the infection with this bacteria. And this is why it's often named an antibiotic nightmare, as I will illustrate later in this talk. Mycobacteria have a very specific cell envelope, which is schematized here. And well, you can identify quite a normal plasma membrane, a periplasm, and a peptidoglycan, which is more or less what you will find in gram-negative bacteria. But this is where the similarity ends, because you can see that the peptidoglycan is linked to an arabinogalactan layer, which itself is covalently linked to a inner leaflet of the outer membrane, which is built of specific lipids, which are called mycolic acid. And the outer leaflet of this membrane is also made of mycolic acids. And then you have the capsule of the mycobacteria, where you can find many strange and specific compounds like mycolic acid, as I already mentioned, but also triallose dimicolate, thiosterol dimycocerosate, and so on. So there are lots of specific compounds in this capsule, and these compounds are essential to the bacterial survival, and in some cases they are also essential to the pathogenicity of the bacteria. And of course, this huge envelope is highly hydrophobic and explains why most of mycobacteria are highly resistant to antibiotics. If you look on the metabolic pathways that are responsible for the biosynthesis of these compounds, these metabolic pathways involves many enzymes which are called megasynthases, such as fatty acid synthesis, PKS for polyketide synthesis, or NRPS for non-ribosomal peptide synthesis. And this for instance, if you take the fatty acid metabolism, they are synthesized de novo by a normal fatty acid system synthase like you can find in most cells, eukaryotic and prokaryotic cells, which will be able to synthesize short-chain fatty acids, which will contain about up to 24, 26 carbon atoms. And these short-chain fatty acids can be further elongated by another system, fatty acid system, which is called a fatty acid system 2. And this is not a mega enzyme, it's just a huge complex made of many enzymes that will elongate the short-chain fatty acids up to a chain length of about 60 carbon atoms. Then those short-chain fatty acid and a long-chain fatty acids are condensed by this enzyme, PPTase in order to build mycolic acids like this, which will be found also in other compounds, in more complex compounds. A Fas2 system is very specific to mycobacteria, and it's already targeted by many of the antituberculous compounds. All this fatty acid system, this PKS, these NRPS enzymes usually display an iterative mechanism where multiple reactions are sequentially produced in order to deliver the final product. So for this reaction to occur, the growing substrate has to be shuttled between the different catalytic domains of the megasynthesis, and this is the function of a specific domain of the enzyme, which are called the carrier protein domain, which I'll identify as CP domain later. And this is where the enzyme we are interested in, the PPTase, step in. PPTase catalyze the transfer of the PPent, the phosphopantetheinyl arm of coenzyme A, to a conserved serine residue of this carrier protein domain, and this reaction occurs in the presence of divalent cations. So this post-translational modification allows the tethering of substrate and chemical intermediates and transfers them from different catalytic centers of the various megasyntheses where the enzymatic modification can proceed. Carrier proteins are essential and mandatory component of all megasyntheses, and the enzyme, all these megasyntheses depend on two enzymes, PPTase, in order to function in mycobacteria. One of them is called ACPS, which is responsible for the activation of the fatty acid system specifically. And the other one, which is called PPTT in Mycobacterium tuberculosis and PPTAB in the case of Mycobacterium abscessus, is responsible for the activation of all about 20 polyketide synthetases in Mycobacteria and a few NRPS. So the group of Christophe Guillot here at IPBS showed that these enzymes were essential to the growth of the bacteria in vitro, but also in some infection model. And if you're able to inhibit PPTT in Mycobacterium tuberculosis, this results in the inhibition of all these PKS enzymes being not able to produce most of these compounds, and this leads to the death of the bacteria. So in our case, PPTAB seems to be quite a promising way to fight against Mycobacterium abscessus infection, as it should result also, the inhibition of this enzyme should result to the inhibition, a similar inhibition for the synthesis of this compound, and likely to the death of the bacteria. So previously, so in order to follow the reaction of this PPTase, there is an easy enzymatic assay that can be performed, because when the PPent arm is transferred to the serine residue, you can see that you introduce, well, it's missing on the slide, but it's actually there. You introduce a negative charge, an additional negative charge, which will modify the way this enzyme migrates on a urea page gel. And so you can see the apparition of the second bond, which corresponds to the activated CP domain. If you add DMSO, which is often used as a solvent for compounds, you can see that the reaction proceeds more or less normally. But if you add an inhibitor, you can see that there is no more a transfer of the PPent arm onto the CP domains, and you can therefore see that the enzyme is inhibited. I previously mentioned that Mycobacterium abscessus was called the antibiotic nightmare, and this is due to a very low susceptibility to antibiotics, and it's even lower than most of the mycobacteria, such as tuberculosis. This can be illustrated by the action of a specific compound, which is called the compound 8918, that was identified by the group of Ballinger about 2 years ago. And you can see that this compound binds to the enzyme PPTT, so the PPTase from Mycobacterium tuberculosis. It binds into the active site, and this results in the inhibition of the enzyme. As you can see, in the presence of the compound, the PPTT enzyme is no more able to transfer the P-point arm to the CP domain. And what is good in the case of Mycobacterium tuberculosis is that the inhibition of the enzyme translates into the inhibition of the growth of Mycobacterium tuberculosis. We checked that in the case of Mycobacterium abscessus, if you take the PPT-AB enzyme, so this is an overlap of the structures of the PPTT enzyme in gray in the presence of the 8918 compound overlaid on the structure of the PPT-AB, so the enzyme of abscessus in the presence of the same compound in green. And you can see that the binding is highly similar in both cases. And as expected, this translates into the inhibition of the enzyme in an in vivo assay. But what is bad is that it does not translate into an inhibition of the enzyme growth. You can see that even with high concentration of the compound, the growth of the bacteria is barely affected. So this compound is not able to cross the envelope of Mycobacterium abscessus, although it is able to cross the envelope of Mycobacterium tuberculosis. So this warns us that targeting an enzyme of Mycobacterium abscessus and PPT-AB, for instance, will be quite difficult. But we nevertheless set up a large project to design and identify inhibitors of several PPTase, so the PPTase enzyme from tuberculosis, the PPTase enzyme from abscessus, but also the enzyme from Pseudomonas aeruginosa, which is also involved in the infection of lungs infection in cystic fibrosis patients, and also enzymes from some pathogenic E. coli strains. And our project was to, using a massive screening approach, we identify and design new inhibitors with the goal, of course, to try to obtain new antibiotics or antivirulence drugs. Today, I will not speak about the whole project. I will just focus on what we've done on the PPTase enzyme from Mycobacterium abscessus. So the whole project combined high throughput screening on whole cell-based assays, virtual biophysical screening of several libraries, a small library we have in our platform of about 11,000 drug-like compound, and a smaller library with fragments, about 940 fragments. And this project also involved the structure determination of all these enzymes and chemical synthesis and optimization. But today I will just focus on a few points of this project. It's the screening of this fragment library using biophysical methods, and the structure determination of the complex of these compounds in the presence of the enzyme. So, as has been shown already yesterday, fragment-based ligand discovery has many advantages, especially for an academic group. Yesterday, there was a talk about the chemical diversity that was possible to achieve 4 molecules with about 14 to 15 non-hydrogen atoms. And this chemical diversity is reasonably large, and with a library, a fragment library containing about 1,000 compounds, it's quite already a decent exploration of this chemical diversity. With drug-like compounds, even with millions of compounds in your chemical library, you would not explore the possible chemical diversity as efficiently. From a purely financial point of view, it's of course much cheaper to build 1,000 fragment libraries than building a 1 million drug-like compound. Fragments are very good also at exploring potential binding sites. They usually bind to the binding site with a high efficiency, although it is with a very low and very bad affinities. So as the affinities are not good, you will need dedicated methods in order to detect binding, but because it's very likely that in a normal enzymatic assays or cellular assays, you won't detect any inhibition with so low, so bad ligands. So if we look at our library, so as I already mentioned that it contains about 940 compounds. They are available in 96 well plates as a 10 microliter solution of 100 millimolar in DMSO. Here you have a histogram of some of the properties of the fragments in this chemical library. You can see that their average molecular weight is about close to 200 Dalton, 13 non-hydrogen atoms, 1 or 2 hydrogen bond donor, etc., and computed solubility, which is about 0.7. And what we checked experimentally that the solubility was indeed enough using NMR before performing our screening assay. So for screening the picked library, so we decided to follow a rather classical screening approach, where we wanted first to identify which fragments were actual ligand or binders to the protein, and this was performed using differential scanning fluorimetry, and we wanted to only use X-ray crystallography on the fragments that were identified by DSF. As you know, thermal shift analysis, you follow the fluorescence of a probe while increasing the temperature of the protein. And when the probe is in a hydrophilic environment, it's not fluorescent. And when the protein denatures, the environment of the probe becomes more hydrophobic, and you have an increase of the fluorescence. If you take the derivative of this curve, you can easily see the midpoint of the denaturation curve, which is called the melting temperature. And you expect that if you have a protein with a melting temperature of something like 44, 45 degrees as here, when you add a compound in the medium which does not interact with the protein, it does not change the TM. But when you have a compound that interacts with the protein, you expect to have an increase of the melting temperature as the formation of the complex stabilizes the protein. And when you have identified all the binders, since DSF is only able to tell you it's likely to bind, but does not tell you where the fragment binds and what is the pose or what is the structure of the complex, does not tell you anything about affinity. We wanted to go further with the identified binders using X-ray crystallography in order to determine the structure of the complex, which will confirm the binding and which will be of great help when it comes to the fragment optimization in the hope of going toward drugs compound or drug-like compound. So we performed these experiments on all our fragments, and we had some fragments which, as expected, induces an increase of the melting temperatures, and some of them which induces a decrease of the TM, so it destabilizes the protein. And this is not uncommon. It always raises the same question: what should we do with these fragments? Should we try to optimize them, or should we just forget about them? So if you take the whole result from screening of the 940 compound, you have about 20% of the compounds which induces an increase of the TM by more than 2 degrees, 3% of the fragments which induce a decrease of the melting temperature of about 2 degrees, and in 5% of the case we were not able to measure a TM. So this means that we have about 225 fragments which are supposed to interact somehow with our protein. So this is quite a high hit rate, but we were not really confident with these results because, of course, you do some control experiments for each plate. You have several where you only have the protein in your plate when you perform the DSA, the thermal shift analysis, and we observed quite an important variation of the reference melting temperature of the enzyme alone. You can see that the TM, the melting temperature, was going, depending on the well, from 46 degrees to close to 50 degrees. So it's nearly a 3 or a 4 degrees variation of the TM. Even if we compare the values measured from adjacent well in the same plate. So we were not really confident about this, the significance of this TM variation. So instead of relying on these results, which were a little bit fragile in our opinion, we just decided to screen the whole library using crystallography. So of course, this implies that you will have to determine several hundreds of crystallographic structure, and you have to carefully think about the way you will prepare the crystal of the complex. So we decided to use a method which is called the dry coating of plates. This means that you will dispense on the crystallization plate about 1 microliters of your fragment in the MSO. You will leave the plate for evaporation under the fume hood for a few days. And when the compound is completely dry, you dispense on the dry compound the crystallization solution, and you let crystallization occur. In our case, in about 91% of the cases, we managed to get crystal obtained in the presence of the fragment. So this is called co-crystallization experiments. Of course, it's not because you have a crystal in your drop that it means that it's a crystal of the complex. It can also simply be the crystal of the protein alone. Then comes the difficult part, because we had about 850 crystals. You will have to cool them in liquid nitrogen in a cold room, so it's rather cold in there. Then you send them to synchrotron. So we mostly use the automated massive beamline at the ESRF in Grenoble, but we also use beam time at Alba in Spain and at Soleil close to Paris. And we managed to collect about 840 data set, which means that it corresponds more or less to 7-day use of beam time, of continuous use of beam time. And it allowed close to 700 data sets to be collected, which translated into 680 structures. At this point, you have to make a decision, because you have a lot of structure and you don't want to spend time to refine structures where the ligand, the fragment did not bind, and you want to concentrate only on the structure where you have a fragment present in somewhere on the structure of the protein. And what you have as an information to decide whether the ligand is in the structure or not is the electron density map. Some examples are shown here. So you have what we call a 2FO minus FC map, which shows you the electron density map of whatever is in the crystal. And more interesting is what we call a difference map, which will show you the difference of the electron density between your model. The model is currently the enzyme without any ligand, and what you have in the crystal, hopefully a complex. So what you hope to see there is a large green peak showing you that a fragment was bound at this place. And as you can see here, there is no such peak visible. And this is very difficult to identify when the ligand is there, is it present in the structure with a low occupancy. Hopefully, Peace and colleagues from Diamond's synchrotron imagine the procedure that they call the PANDA, that was designed to allow to identify weakly bound fragments in crystallographic structures. So if you apply to this procedure to the same data set as the one shown here, you can clearly see that there is a signal which indicates that a fragment is actually bound there, even if the normal electron density map does not say so. So this allows to identify among your 600 or 700 data sets which are the ones you should concentrated on in order to pursue the refinement, and which are the ones you can just forget about. So these procedures, this PANDA procedure allowed to identify about 70 positive hits, so it's a 7.6% hit rate, and all these compounds you can see there, and you will see it more clearly in the next slide, that they accumulate at 2 binding sites on the protein. One of the binding sites is very close to the cofactor, so the coenzyme A, which is quite interesting because it means that we are close to the active site. And the other binding site, you can more or less guess it behind this image. If I turn the image, you can see it there. It's not that interesting because it's a surface binding site, and it's a binding site which is at the interface between 2 neighboring proteins in the crystal. So it's very likely to not exist in solution. So I will only focus on the substrate or the coenzyme binding site. So next step is trying to optimize these fragments in order to have compounds which hopefully will display some activity in the enzymatic test at first, and later on bacterial culture. So the classical way to do this is to take the structure of the complex, to focus on the interaction area between the fragment and the protein, and try to see which are the characteristics of the area where the fragment binds, and see that you have somewhere a lipophilic place, some places here where you have a donor acceptor of hydrogen bonds, an oxygen atom there, and a lipophilic tunnel going there, and an area where you're close to the opening of the active site, where you can imagine placing many different things. And from that, you can imagine many different chemical modification of the fragments, and this is where the chemist will start to play and synthesize lots of compounds. But this is only this is starting, for instance, from one of the fragments which was identified as a ligand. But as you can see, we have many of them. There are lots of fragments there. And the question is, which one would be the best as a starting point for the optimization process? So one way, since we are not able to measure any binding affinity, to measure any enzymatic inhibition or whatever, we cannot rely on these experimental ways to identify the most interesting, most promising starting compound. So instead of that, we focused on the mechanism of the reaction. So the reaction is the transfer of the P-pont arm from coenzyme A to conserved serine residue on the ACP, so the carrier protein domain. So you can see from this schematic that the reaction starts by the deprotonation of this serine by glutamic residues, and the deprotonated serine will then attack the phosphate group of the PPANT arm and release the ADP compounds. So this glutamic 153 is represented here in the structure. You can see that it's in close interaction with divalent cations. It's magnesium or manganese. And you can also see from this mechanism that the position of these manganese and the interaction of these ions with the proteins are likely to be quite important for the mechanism of the reactions. So you have this glutamic residue which interact with this manganese ion there, and you also have this histidine residue which participates to the positioning of these second ions there. Something which is also important for the positioning of the proper positioning of the cofactor in gray is these arginine residues which will make salt bridge interaction with the phosphate group of this cofactor. So we will focus now on these three residues, and on the next slide, on this slide, what I've done is I've superimposed all the structures of the complexes with a fragment bound in this coenzyme I binding site, and I just show a few side chain in this binding site. And you can see that the bindings, the side chains are very conserved across the structure. You can see that this lysine does not has a single conformation in all structure. It's the same for this glutamic residue, for this one, and for the arginine over there. But if you look at the three residues we identified on the previous slide from the mechanism, you can see that in some structures, the glutamic 153, which usually interacts directly with these manganese ions, actually interacts with the fragment instead of interacting with the cations. It's the same for this histidine residue that you can see in one of the structure. This histidine residue is no more involved in the binding with this manganese, but is shifted outward, and it's the same for this arginine residue, which normally interact with this phosphate group and contributes to the correct positioning of the cofactor. In some structure, the conformation completely change and does not interact with this coenzyme anymore. So it's come to the point that in some cases, the fragments will decrease the interaction of the protein with its cofactor. And it's to the point that in some structure, it seems that the electron density suggests that the cofactor is actually not present in the structure, as if the fragment binding would expel the cofactor out of the binding of its binding site. And this, of course, Laurent, is quite interesting. Yeah.
You have 5 minutes left.
Yeah, it's my last slide, so it's finished. So it's perfect. So it's really interesting from the inhibition point of view, because it means that if you have a fragment, all the fragments bind in this area. If you have a fragment bound there, it will prevent the substrate from coming into the active site. But in some case, it might also induce the disappearance of the coenzyme A cofactor from the enzyme. So it should result in a double inhibition mechanism, which would be quite interesting from the chemical point of view. So we are working along this line, and we would be really happy to test all the nice tools that Marcus showed yesterday in order to try to improve our fragments and to be able to obtain compounds, hopefully with inhibiting activity. So this project involves quite a lot of people in our institute, IPBS, people from the group I'm working in, with the people in bold, which are really a lot involved in the project. people from the NMR group and people from the molecular mycobacterial pathogenesis group. And also we worked very closely with chemists in order to synthesize the optimized fragments. So thank you for your attention, and I would be glad to answer any question if there are any. Thank you.